You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast, and I have Katie Peichel, formerly Catherine Peichel, head of the Evolutionary Ecology Division at the University of Bern. Actually, it's the Institute of Ecology and Evolution. So, Katie, thank you for coming. How are you doing?
I'm good. Thank you for having me.
Yeah, you appear to be specialized in the exact area I've been asking about for a long time. Is how do new species occur? The only thing I've ever heard is, they occurred millions of years ago and no one's ever seen them. And, I don't quite believe that. So I always want to know, how do new species occur? What pressures and what evolutionary changes do creatures go through to become a new species? So hopefully you can shed some light.
I hope that I can. I think it's still quite a mystery, but we're starting to gain some more insight into that process. So,
okay. Yeah. Before we begin, I hear phenotype, that word used a lot. Can you describe what it means to you so that when we start using it and you start using it, that it makes sense to listeners?
Sure. A phenotype is anything that we can measure on an organism. So that could be height or weight or lifespan or a particular behavior. So we often use the word phenotype in the same way as trait, but it basically means anything we can measure.
Okay. Well, very good. So what's your current state of understanding of what constitutes a species? It seems like, when something's a different species, species A and species B, they can't breed to create viable offspring. That's the definition I've always heard. But what is your definition of a species?
So I think I would say this is still somewhat debated by biologists, how exactly to define a species. And for me, I use the same one that you just mentioned, which is called the biological species concept. It's first put forward by Ernst Mayr, and it basically says that species are groups of actual or potentially interbreeding. They're groups that can interbreed with each other, but they're reproductively isolated from other groups. So they basically can't breed with other groups. And that could be because of they just don't breed in the same place or at the same time. That could be because they have behavioral differences that prevent them from breeding with each other, or that could be that they might breed with each other, but the hybrids that they make are sterile or they're inviable. So that's kind of
a donkey or a mule, I forget which one, but
exactly.
Two different.
Or things like,
so is that considered a new species, or is it not?
Well, it's not really a new species. That would be a hybrid between two species that is not really going to reproduce on its own. So it doesn't form its own species. So the key to this definition of a species is this idea of reproductive isolation. So barriers to breeding between two groups.
So, have we seen speciation occur, while people have been observing in the last 1,000 years or so, a couple thousand years?
Yeah. So, we can find groups that are very, that we sort of call different species because they are reproductively isolated from each other and they're very young. So, for example, I work on these groups of fish, this group of fish called Sticklebacks. And within a few lakes in British Columbia, you actually have two species of these stickleback fish. one that's living in the very open part of the lake. Those fish are feeding on zooplankton. They're very they're small, they're slender, they have a lot of physical changes or phenotypes that adapt them to living in this open water habitat. And then within the same lake, you have fish that we call benthics, and they're living in the very vegetated part of the lake. They're eating large macroinvertebrates. They're quite large and stocky fish, and they have phenotypes that adapt them to eating these very different kinds of food that like these small invertebrates in the benthic environment. Now, these two types of stickleback that live within the same lake, they meet each other so that you can find them breeding the same time and in the same location, but they actually don't breed with each other because they have behavioral preference for their own type. So limnetic females like to mate with limnetic males, and benthic females like to mate with benthic males. And so essentially they are two different species, even though these lakes were only formed about 10,000 years ago. So we know that when there was glaciers covering the northern hemisphere, covering British Columbia, there weren't lakes there. These freshwater lakes were not present, and they've formed since then. So those are relatively young species, at least on an evolutionary time scale.
Can they breed with each other? They just prefer not to, or they literally can't?
That's exactly right. So they can breed with each other. So if we bring them into the lab and we take sperm and eggs from the benthics and limnetics and we mix them up, we can get viable hybrids, and those hybrids are fertile. They can have their own offspring. So they're physically they're able to reproduce with each other, but they just prefer not to.
So when is also the ancestor of the two living in that same lake, or is it gone?
So we don't know. So the ancestor of most freshwater stickleback populations, we think, is a marine stickleback, which when the glaciers melted that were covering most of the northern hemisphere about 12,000 years ago, these newly created freshwater lakes and streams were invaded by marine sticklebacks. And so it's thought that marine sticklebacks gave rise to these benthic forms and these benthic species and these limnetic species. But what's unique about these two these lakes where we find the benthics and limnetics is that there's geological evidence that those lakes were invaded twice by marine ancestors. So there was an initial founding or invasion of the lake by marines. It's thought that those fish became the limnetic species. And then, I'm sorry, I said that the wrong way. It's thought that the first invasion became benthic. And then there was a second invasion of marine sticklebacks, and those, because this benthic habitat was already taken, moved into the open water and became the limnetic fish.
So what do you think leads a creature to change over time, to evolve and to eventually form a new species? Is it, some theories, maybe my own, is the piling up of epigenetic changes that slowly cause the underlying DNA at some point to change, or just the epigenetic changes lead to a different phenotype? What do you see?
So in the case of these sticklebacks, our idea is that it was really adaptation to these very different environments. So the benthic sticklebacks were kind of changing in their phenotypes to live and take advantage of these benthic resources, macroinvertebrates. The limnetics also were changing over time to adapt to eating these zooplankton, and there was accumulation of genetic changes in the benthics and the limnetics that then led to this what we call behavioral isolation. And so we really think in this case, speciation was a byproduct of adaptation to these different environments. And in fact, the traits or the phenotypes that make a benthic female choose a benthic male, so this big, deep body, are really the same traits that make them able to forage or eat in these very different habitats. And so it was really just a kind of a consequence of adaptation to these very different habitats that led to this process of speciation.
But when you say adaptations on what level specifically do you think is happening?
We were thinking about adaptation on the level of the when we talk about adaptation, we talk about phenotypes that make an organism more fit in their environment. So what that means is that they survive and reproduce better. And so you have these phenotypic adaptations, phenotypes that increase your fitness, but then there must be genetic changes that underlie those phenotypic changes that cause these adaptations. And so we really are, in my group, we're thinking about trying to understand what are the phenotypes that are important for adaptation and therefore for speciation, and then what are the underlying genetic changes that allow these sort of phenotypic changes to happen.
Yeah, that's interesting. I guess there could be a compounding of phenotypic changes by various mechanisms, but it's an adaptation to the environment, which makes total sense to me. Mm-hmm. But it's funny. I wonder if what causes the sexual reproduction isolation is the preference, the mating preference, just like in the Sticklebacks. If at the end, that's what really finishes it off and finishes diverging the species.
Well, we think, at least in these Sticklebacks, I think about these behavioral differences as an important component. So it certainly kind of reinforces, which is, I don't want to use the word reinforcement because it has a specific meaning, but it basically helps them maintain themselves as separate. But this behavioral isolation is not perfect. So sometimes a limnetic will mate with a benthic or vice versa. And so you still get some exchange of genes between them. And what we tend to think about in the speciation field is that things like intrinsic hybrid inviability is what kind of completes speciation. So that would be if hybrids are formed, they just die. And we don't see evidence for that in this system. If we make, as I said before, if we make the hybrids in the lab, they can actually survive just fine. In the lab where we give them food and we give them sort of nice environment. It is true, however, that these hybrids in the wild, when they're formed, they're not very fit. They can't they can't forage very well in the normal environment of the lake, and so they tend to die as well. But a few hybrids every year, some percentage of hybrids actually are formed and do actually survive. So that's why we call these sort of incipient species. They're in the process of speciation, but they're not quite full species yet.
It'd be interesting if you could do an experiment where you released hybrids into the lake and watched what they did over several generations. If they moved to the benthic form or the other form, or to a third form,
Yes, it would be really cool to do that. We cannot do that in nature just because we don't want to do those sorts of experiments and mess up natural lakes. But I've worked quite closely with Dolph Schluter, who has a lab at the University of British Columbia in Canada. And Dolph has this amazing series of ponds in which he has kind of replicated the environments that are present in the natural lakes. So these big ponds are about 20 meters by 20 meters, and they can hold thousands of sticklebacks. And there's a benthic part of the pond and a limnetic part of the pond. And we can put hybrids in there and we can follow them over time and over generations and see kind of who survives best in the ponds. And that's how we know, for example, that some hybrids do really badly. They can't really eat either sort of parental food. They can't eat the zooplankton that the limnetics eat, and they can't eat these macroinvertebrates that the benthics eat.
So when does nature create viable hybrids versus non-viable? Is it when we force it? It seems like they're non-viable, but when nature does it, though we haven't observed it directly, it seems like they're viable.
Yeah, that's a good question. I think it's partially there's a little bit of a bias, right, in what people take to the lab and try and cross together, and what people have actually, what species people have actually looked at in nature. I would say most of the things that we as humans recognize as good species. In fact, if we did this test and we tried to make hybrids, generally the hybrids don't do very well. But there are more and more of these cases of, like, my sticklebacks and in many other systems where there's things species that look different from each other, but aren't completely reproductively isolated from each other. And it's only now, I think, that biologists are looking more closely, and it really takes someone who's very careful and thoughtful and goes out into nature and looks into a population and says, that's interesting. Some of these fish look like this, and some of these fish look like this. I wonder if they're reproductively isolated from each other, and then starts to do these tests. So I think there's more and more interest in looking at these cases like the sticklebacks of these sort of young species, and we're discovering them more and more all the time, the more people look.
Are you able to guess how many generations it takes for speciation to occur? Is that at all? I mean, we have so far the upper limit or the lower limit is 10,000 years, I guess. But
right.
How short could it be?
Well, I think it could be shorter, and people have done some experiments, for example, in the lab, where they have selected, I'm not going to get the details of the experiments right, but for example, you can select things like yeast growing on different sort of resources, so selection on different habitats, and in just a few dozen generations evolve reproductive isolation between them as a consequence of adaptation to divergent habitats. So I think it can happen pretty fast. It's just a question of us finding it.
Well, yeast, I guess, we can call them simpler organisms, but more complicated organisms. Have we been able to try it with fruit flies?
Yeah, exactly.
Pretty fast.
Yeah, there's some experiments as well in fruit flies where if you adapt fruit flies to different habitats, you can evolve some forms of reproductive isolation between them, again, in not very many generations.
What's the approximate of not many, like 100, 1,000?
Yeah, I would say on the order of, tens to 100 generations.
Yeah, that's pretty short.
Yeah, it is pretty short. I think an important question that many of us in the field of speciation are trying to understand now is it seems relatively easy to get these reproductive isolation to evolve in these kind of young species pairs, but how often do they persist? So how do we connect these patterns of these very young species pairs that we find to kind of the big patterns of diversity that we see in nature, where there's all these different species, and what causes them to persist and to remain as separate species is a question that I don't think we have an answer to.
Well, I think evolution appears to happen in response to environmental stress and environmental changes. So as long as those persist in some way, even if the environmental stress changes at a certain point, the species doesn't seem like it goes backwards. It'll continue forward along a new path.
That's right. And I think the question is, how long does that part take to happen? So again, as an example from sticklebacks, I told you about these species pairs in these lakes in British Columbia. Well, we have evidence that in one lake called Enos Lake, the species pairs have actually collapsed. So there was an environmental change to the lake, and there used to be benthics and limnetics in that lake. They'd been quite well studied. And then there were some environmental changes to the lake. And now they're hybridizing with each other. And so we don't see distinct gene species anymore. They've collapsed back into what we call a hybrid swarm. And so that's a case where the species did not persist. We also have examples, or kind of a question is, we have these young species in sticklebacks, which are just 10,000 years old or so. And as I told you, they don't, if we breed them in the lab, we get viable hybrids, and the hybrids can have offspring. But then we have some species pairs in sticklebacks that are like about 1 million years old. And there, if we breed them in the lab, the hybrids are sterile. So the males don't make sperm. And so there's this gap between this 10,000 years, you can kind of evolve species, but it's not complete. And then we have these sort of older species pairs that are 1 million years old or so. And there you get sort of more complete reproductive isolation or speciation. So we don't understand what's pushing things. Kind of, is it just time? Or are there other events that we don't know about?
What happens when you run into a neo-Darwinist? knowing what you know and experiencing what you've experienced?
Um, yeah, it's interesting. I usually, when people ask me what I do, and what I'm interested in, I usually tell them I'm really interested in the genetic changes that underlie why organisms be different from each other or behave differently from each other. So I try and talk mostly about kind of what I do from that perspective and convince people that that's interesting, and then kind of tell them, well, this is evolutionary biology.
So you're looking at the phenotypical expression of these creatures, but what about the underlying genetics, the epigenetics? Are you looking there, or is that too much work and other scientists have to do that?
No, that's a huge part of my research. So these fish that I study are a really great system for that, because as I mentioned, we can take these species back to the lab and they do breed with each other. And we can breed them in the lab, and we can use genetics to understand, well, what are the actual genes that underlie these phenotypic differences between species, particularly the phenotypic differences that lead to reproductive isolation. And so my lab has done a lot of work to try and understand what are the changes in the genome or the DNA sequence changes that actually contribute to these phenotypic adaptations and to speciation. It is a lot of work, but...
Yeah, well, what are you seeing? I mean, what seems to be, is it various mechanisms? Is there a predominant one that seems to happen early on or later on?
Yeah, there's I think there's a few lessons that we've learned so far from these sticklebacks. And so the first is a question that's been debated really since the time of Darwin, which is, how do phenotypic changes happen? Is it because there's like a few mutations that have huge effects on phenotypes, and so you kind of get these big leaps of evolution? Or does evolution happen by many mutations that each have relatively small effect on phenotype? And we've been able to show that it's a combination of, there are some genetic changes that have relatively large effects on phenotypes, and there are some changes, then kind of more changes that have smaller effects on phenotypes. So just change the phenotype a little bit, but you get this nice distribution of effects. And that's important because people really thought for a long time that the mutations that led to phenotypic adaptation and to speciation would be so small that those mutations would have such minor effects on phenotypes that you would never actually be able to find them. And so we've shown that you can actually find these genetic changes that are important for evolutionary changes in phenotypes.
So they're not small. How appropriate are they? How smart are they? How smart are the changes? Do they appear to be just random and some of them happen to work? Or do they appear to be very guided and focused, as if the organism is adapting?
In our evidence, we can't necessarily say kind of the mutational processes that led to these changes. They don't. It more appears to be there's mutations that happen, and then selection acts on those, on the phenotypes, and if the phenotype causes an increase in fitness, then they're under selection. One interesting thing, though, about sticklebacks, as I mentioned earlier, there's these marine sticklebacks have moved into freshwater, that's what it has established freshwater populations, which is where we see all of the sort of diversity in phenotypes. And these marine sticklebacks are still moving into freshwater to breed every year. They're like salmon; they move into freshwater to breed, and when they do that, they come into contact with freshwater resident sticklebacks. And so there's a little bit of hybridization that happens, and the genes or the alleles of those genes, the forms of those genes that are kind of adaptive in freshwater, get into marine sticklebacks. So if we take a bunch of marine sticklebacks and we sequence them, we can find that some of them at very low frequency have an allele that is good in freshwater. And so what we think is happening is that when marine sticklebacks go into freshwater, they invade a new freshwater population. Those freshwater alleles that are sort of hiding in the marine population get selected upon and rise in frequency in the freshwater populations. And so if we go to 100 different freshwater populations across the world, we find that they have the same freshwater adapted alleles because the marines are kind of, we really, and sticklebacks are not really waiting for new mutations to happen, but rather these are old mutations that have happened. Selection has favored them, and then they've kind of stuck around in the stickleback population. And then when there's selection for a particular phenotype in freshwater, the variation or the mutations that are needed for adaptation are already there.
But when the marine sticklebacks go into freshwater, are they mating with freshwater sticklebacks, or are they mating with other marine sticklebacks in the context of the freshwater?
They're mostly mating with other marine sticklebacks, but occasionally they do mate with sorry, I hope I just said that right. The marine sticklebacks are mostly mating with marine sticklebacks, but occasionally they do mate with the freshwater sticklebacks, and that's how this exchange of these alleles is happening.
I mean, it could be two things, though. If they're mating in the freshwater context, that could temporarily be changing their gene expression. They mate, maybe the gene expression is heritable. Now the new marine that comes out of that is changed just because it happened to be conceived in a freshwater environment, you know?
It could be. We currently don't have any evidence for any sort of epigenetic changes like you're talking about in sticklebacks. We have very, very good evidence for DNA sequence changes between marine and freshwater sticklebacks that kind of differentiate them.
I would think the marine sticklebacks, once they go into the freshwater, their gene expression would change dramatically. It's a very different environment, at least while they're in the freshwater environment.
That's right.
Have you observed that?
It does. So there are people who've done experiments where they put marines into fresh water, and there are changes in gene expression. But to date, there's no evidence that change in gene expression is heritable. So the only evidence we have for sequence changes that are heritable between marine and freshwater sticklebacks are kind of these DNA sequence changes that differ.
What percentage of the marines breed with the fresh, and does that change the fresh population over time as it gets invaded over and over every year?
That's right. It's a relatively low percentage of hybridization between marine and freshwater populations. It varies from place to place, but it's generally just a few percentage. And usually those hybrids don't do particularly well, but it seems to be enough hybridization that the marines kind of pick up these freshwater adapted alleles and then are carrying them around.
So why not do an experiment with marines that have no freshwater alleles? Put them in a freshwater environment, see if you can breed them, and then see what happens to them if they start accumulating those changes.
That's a great experiment. So it's sort of being done soon. So in fact, right now in Alaska, some colleagues of mine are putting marine sticklebacks into some lakes where all of the fish were killed. And so these are freshwater lakes. And so they can put marine sticklebacks into those lakes and kind of see how they evolve over time.
Okay.
What would be cool, what you're suggesting, so probably these marine sticklebacks that are going to these freshwater lakes are going to have already some of these freshwater adapted alleles because of this, what we call standing genetic variation in the marine population. But I think what you're suggesting is that we would take marine sticklebacks that we know don't have any of these sort of freshwater alleles already to see kind of what would be the role of new mutation in an adaptation to freshwater. So that'd be really a cool experiment if we could have two populations of marine sticklebacks invading, kind of going into different lakes and asking, do the ones with no sort of existing freshwater alleles, how do they adapt and follow those over time? That'd be an interesting.
Yeah, I mean, from what I've heard, I mean, gene expression can change incredibly quickly. sometimes within like hours,
Yep.
or minutes or hours. So, I mean,
Yep.
I don't know. Interesting.
Yeah. But again, the question is then how does that then get transmitted to the next generation? So the gene expression changes, that just changes on the RNA level, but how is that actually transmitted? So an
or is
interesting
it
question.
transmitted, right? Yeah. So what are some upcoming experiments or things that you want to figure out? What are you working on this year?
Let's see. So I'm involved in this big experiment in Alaska, not so much the marine sticklebacks going into freshwater, but we're putting some sort of these benthic and limnetic type of sticklebacks into 8 different lakes. And we want to understand kind of when, again, the question like when these different fish go into new environments, what are the changes in the genome that allow them to adapt to these new habitats? But we're also interested in understanding kind of the effect of the sticklebacks themselves on the whole ecosystem. So how do they change the ecosystem of the lake? And then do those changes in the ecosystem sort of feed back onto the sticklebacks? And do they change phenotypically over time? So this is a big sort of experimental evolution that people traditionally do in microbes because, or fruit flies where you can get microbes like yeast or bacteria, you can get hundreds of generations really, really fast. In Drosophila, you can have, 4 to 5 generations, 10 generations a year. Stickleback generation time is 1 year. So this is a very long-term experiment that's just getting started, but we look forward to kind of seeing how that literally evolves over the next 10 years or so. And then I'm really interested in kind of understanding more about kind of changes in the genome itself and how changes in the genome, so the structure of chromosomes, how does that affect adaptation and speciation?
Okay. Do you know anyone that, actually, before I get to that, the person that has the pond experiments, I would think they could create ponds of different salinity, different kinds of conditions, different temperature and all that, and keep that pretty consistent. So I mean, you can see the degree to which, the sticklebacks changed, whether the change in their current conditions is slight or great, and that may cause speciation faster. I mean, at a certain point, it'll kill them. How far could you push it and see fast speciation? Fast changes.
Yeah, no, that would be an interesting. So the ponds we can't control so carefully, temperature and salinity, but those are the kinds of things that that would be fun to do. Yeah.
Okay. Do you know anyone that's working on this kind of stuff in people, not causing it to happen, but observing it? Are we observing in the human population significant phenotypic changes over time?
I mean, I think people are interested in looking at different populations of humans that have phenotypic differences and then understanding, what are the genetic changes that underlie those phenotypic differences, and also what are the sort of selective forces, ecological forces that lead to those changes. So, for example, people working on adaptation to high altitude in Tibetans or in people in South America, those are the types of, I think those are very similar sorts of questions to what we're doing in sticklebacks.
Okay, well, very good. So what could happen in the next few years that would delight you, that you'd be really excited to see as a result?
Whew, that's a good question. I mean, for me, what we still don't know in almost any case in evolutionary biology is, let's say we have a phenotype that changes. What we really want to know as an evolutionary biologist is what are the genetic changes that underlie that phenotypic change? So what are the specific DNA sequence changes? What are the genes? And then we also really want to know what are the selective forces. So what is it in the ecology, or why did that phenotype change? Why did that phenotype evolve? And so it's the sort of what we call the proximate mechanisms the genetic changes, and then these ultimate causes, so the selective forces. And I think we really don't have very many examples where we can put all those pieces together, and that's really what I'm trying to do in my fish, is make this really complete story of evolution where we understand kind of everything from the genetic changes to the phenotypic changes to the sort of selective forces. So that's what I'd like to accomplish in the next few years.
Okay. Well, very good. So what's the best way for people to get in touch and ask for questions?
Via email, for sure.
Okay. I guess we'll put that in the show notes. Any other publications people could look at or ways to see the stickleback in action?
I guess all of our publications are listed on my lab's website, so you can include that. I'm trying to think if there's any, and we have some pictures on our website of the fish themselves. Yeah.
Okay. Very cool.
Great.
Katie, thanks for coming on the podcast. I really appreciate it.
Thank you for having me.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.